Autoimmune diseases, which are estimated to affect more than 15 million people in the US, occur when the body responds to immune system false alarms and infection fighting first responders are sent out to attack threats that aren't visible. Scientists have long understood how the false signals are triggered, but the subsequent step of dispatching the immune response is still unknown. At this time, scientists at the Washington University School of Medicine at St. Louis and the Perelman School of Medicine at the University of Pennsylvania have discovered a key component to launching immune activity and overactivity. These researchers identified a protein in cells that triggers the release of infection fighting molecules. The protein provides a substantial target for therapies that could prevent overreactive immune responses that are at the crux of several degenerating illnesses. The study is yet to be published and will be published on March 20th. The team of researchers, co-led by Jonathan Miner, an associate professor of rheumatology and microbiology at the Penn's Coulter Center for Autoimmunity, and David Cast, an assistant professor in the Department of Cell Biology and Physiology at Washington University Medicine, found that by studying a rare autoimmune disease called STING associated vasculopathy with onset in infancy, which is extremely rare, occurring 1 in every 1 million births, leads to the immune response attacking tissues in the lungs and limbs of patients, often resulting in death before adulthood. SAVI, that is the sting associated vasculopathy with onset in infancy, is induced by changes to a protein in cells called STING, which ordinarily acts as a molecular watchdog that responds to the presence of viral DNA by activating the component of the cell that generates immune proteins, which are then released from the cell to signal to the body's immune system to respond to the viral invaders. Sting is overactive, triggering constant immune activity that ultimately damages healthy tissues. Using immune cells that are sensitive to the disease causing mutations in Sting, the team performed a screen to unravel proteins that prevented this sensitivity. One protein, ARFGAP2, is shown to be strongly linked to the final step when immune response proteins get released. This finding was further validated to show that SAVI cells did not produce ARFGAP2 could not, I mean, ensure that STING triggered the release of the immune proteins. It's like a train station, and ART-GAP2 is acting as a conductor, directing which molecules are to be dispatched. If STING and ART-GAP2 are not working together, the trains are stopped. So the team concluded that stopping the never-ending trains in SAVI's constant immune response could be a means of treating the rare disease. They tested the idea in a mouse that was genetically modified to have SAVI, but did not produce the ART-GAP2 protein. They found that the lung and limb-destroying immune response typical of the disease did not occur. So, in effect, if the protein could not be neutralized, the overactive immune response could be extinguished. So, it It is a promising target for a condition that similarly lead to excessive immune response of the same type, such as cytokine storms characteristic of COVID-19 or brain inflammation linked to immune response in Alzheimer's disease. It could open the doors to all these new avenues of potential therapies for many, many different classes of disease, the researchers concluded.